Good morning. Thank you for joining our final webinar of the year, Polymeric and Lipid Nanoparticles for Delivery of Self-amplifying RNA Vaccines, presented by Dr. Viet Nguyen. This again will be our final W.E.S.T Hour due to the holidays. We're always the last Wednesday of the month, and we're looking forward to celebrating Thanksgiving and Christmas. So we thank you for attending. Just a little about our team for those of you that are new. My name is Kim Killian, and I am the Regional Sales Manager for the Southwest. And I work with Viet Nguyen, who is the Field Application Scientist. We're both located in Southern California. And then, of course, our North team is represented by Angel Galarza, who's the Regional Sales Director for the West, the Northwest, and he's located in the Bay, along with Ian Villamagna, who is the Field Application Scientist for the Northwest Territory. And Ian has just recently relocated to the Seattle area. So hopefully you'll see and get to meet Ian if you're presiding in that area soon. And then as always, you can check out our YouTube playlist. It's under W.E.S.T Hour, W.E.S.T W.E.S.T Hour, webinars empowering science and technology for those that don't know the acronym. So check those out. And then I guess finally, we'd really just encourage you to join our final journal club of the year, which is, I believe, it's going to be on November 12th. Our journal clubs, are on the second Friday of every month, and those start at 10 a.m. Pacific time, noon Central. So hopefully you can join us for the final journal club. But we just really appreciate the participation and the support this year. We're really looking forward to continuing this in the new year of 2022. If you have any topics, anything that you'd like to pass on to us, how we can improve these webinars as well as journal clubs, don't hesitate to reach out to any of us. So with that, Viet, I'll pass the baton to you. Thank you very much and enjoy the webinar.
Thank you. Thank you. Good morning, good afternoon, everyone, depending on which time zone you're joining us. And like you mentioned, thank you all for joining. Today we'll be discussing a publication, which is a collaboration from between Precision NanoSystems and Imperial College London, Dr. Robin Shatter's lab, among others. And it's really just a short story on the utilization of self-amplifying RNA vaccine, self-amplifying RNA for development of vaccines. Again, before we hop in, Kim did mention we do have the Q&A options as usual. Please feel free to ask questions also during the presentation if there's something of interest on the slides that come up. We like to start with our vision, which is to accelerate the creation of transformative medicine that significantly impacts human well-being, essentially helping our customers developing their nanomedicines. So to set the stage, we're still during an ongoing pandemic, and what we've learned since the outbreak is essentially that rapid response to outbreaks like this is critical, and in the past year we've seen variants show up, so it's really something that's current and it's needed. And we've also found that really, especially in the initial stages, production capacity was and still is a bottleneck as far as the production of these vaccines and really widespread adoption and vaccination. So it's really important that we have technologies that can allow these things to happen. just take a step back and look at the current vaccine technologies. There is a variety of options. More classically, these were viruses, whole inactivated or live attenuated, or pieces of a virus, subunit vaccines. In general, these have been used for quite a while, and they do work. The challenges come in really the low immunogenicity, so low immune response, and really the difficulty of iteration of these, of manufacturing of these. But there are still, very good technologies. On the right side here, we have couple nucleic acid-based options for vaccines, and these really are widely spread into two major fields of viral vector-based nucleic acid vaccines, which again suffer a little bit from manufacturability or ease of manufacture, as well as potential reuse challenges with reuse, really. And then synthetic carriers, which we'll be focusing on in today's discussion. And these are the ones that are completely synthetic, made of excipients and nucleic acids combined together chemically. And therefore, they're quite easy to manufacture, quite easy to iterate, as showcased really by Moderna, Pfizer, and others during the early stages of the pandemic last year. And it's really what this pandemic has really shown is the power of this synthetic vaccines technology that allowed, again, Moderna, among others, to develop the vaccines, the vaccine within, inject first human patients within 66 days of the posting of genomic sequence. So it's really a powerful technology, is what we've seen since. And to dig deeper into the vaccines, synthetic vaccines themselves, it's really important to talk about the RNA, because not all RNA is made the same. There is obviously different options of the RNA payload that you would use for the vaccines. Some companies focus on really unmodified RNA, and they called it unmodified. They're not base modified, but they're still stabilizing motifs, structural motifs at both ends of the RNA, and it's essentially to enhance the stability of the construct still, as you're working with, because as we all know, RNA is inherently unstable. These unmodified RNAs really, they can be challenging as far as immunogenicity, from the RNA itself. Again, if you're trying to elicit an immune response, make a vaccine, you want to generate immune response from the antigen, say the spike protein for the COVID vaccine itself, not from the RNA, and that's where some challenges come in for these unmodified RNA vaccines. Most of the current approved ones, at least in the US, are base modified. Both Moderna and Pfizer-BioNTech vaccines are base modified RNA, and these essentially are modified to increase translation after delivery, as well as reduce the innate immune response to these vaccines. So these are really more commonly used right now. And then really a newer technology here, self-amplifying RNAs are quite an interesting way of generating really the protein of interest, or RNA of interest, in the sense that it's an RNA that has the ability to self-replicate and therefore amplify its signal and potentially result in reduction in the dose needed to get the same response upon vaccinations. This is a technology that's utilized by, among others, Arcturus, ICL, as well as our own program at Precision NanoSystems. Okay. And to dig deeper, essentially what this self-amplifying RNA technology is, it has an additional really non-structural, well, part of the RNA encodes for non-structural proteins that allow this RNA to replicate and to form multiple strands of this encoded cartridge itself, and therefore essentially amplifying the response. So you need, in theory, less strands of this to get the same amount of strands of your RNA of interest, and therefore the production of your protein of interest. In the case of COVID vaccines, spike protein. So this has a really strong potential in being able to reduce the doses needed, and therefore, reduce the amount of RNA needed in each dose, and therefore make the manufacturing process easier in the sense that you can produce more doses in the same volume. So that takes us to the study that we'll be discussing today. It's currently published in Journal of Controlled Release. First author is Dr. Anna Blackney, who has since moved to UBC. So she was at ICL when and she was driving this study, but she has since moved and joined the faculty at University of British Columbia, and we're looking forward to some more great research coming out of Dr. Anna Blackney's lab. And this is again a collaboration between Precision NanoSystems, titled Polymeric and Lipid Nanoparticles for Delivery of Self-amplifying RNA Vaccines. And the main focus of this study is essentially comparison of two excipients, two delivery systems, if you may. One LNP-based, so this is similar structures to the current Pfizer, BioNTech, the approved vaccines that are deployed and used in humans nowadays, compared to a bit simpler polymeric system. I would say a more classical system, where it's essentially a positively charged polymer mixed together with the negatively charged strands of self-amplifying RNA nucleic acid, essentially forming kind of a ball of yarn with the RNA on it to deliver the RNA of interest. So two structures looking in mice only. So these studies are all done in mice, comparing the two systems, looking at the transfection efficacy as well as functional efficacy, both for humoral and cellular responses. Okay, so the authors start out with essentially looking at the physicochemical characteristics of the particles they've made. In the study, they have screened multiple, essentially a panel of ionizable lipids compared to this polymeric system. And they're essentially showcasing that with this data is that they're comparing apples to apples. They're comparing something of a similar size and similar size distribution, around 8 nanometers, with a PDI of 0.1 or so, with high encapsulation efficiency, so high quality nanoparticles, whether they're these polymeric ones or LNP based. And they also look at the surface charge, zeta potential of these particles, showcasing that indeed the polymeric system, it's a cationic polymer, so you'd expect a positive zeta potential, which they see here. And for ionizable lipid nanoparticles, it's also expected that because the lipids are ionizable, we're looking at a zeta potential that's close to 0 when measured at neutral pH, as far as that. these compositions, these are 4 different lipids, and these tails here essentially indicate, it doesn't indicate anything about the lipids. It indicates the helper lipid used in the composition. The ending with PE here, it indicates that it's DOPE, helper lipid used in this composition. And the PC here is indicating that it's DSPC used as a helper lipid. As far as lipid ratios of these lipids in the LNP composition, it's essentially mimicking the current, it's the same as the current Pfizer, BioNTech, Moderna vaccines, with just the helper lipid swapped out in some of these compositions. And the ionizables here are proprietary Precision NanoSystems ionizable lipids. Okay. Up. So these particles were made with self-amplifying RNA encoding for luciferase in this case, and encapsulated and injected intramuscularly into healthy mice. After 7 days, these mice were injected IP with luciferin substrate, and essentially the authors used IVIS to assess the expression levels, the production levels, in this case, this reporter gene luciferase, upon injection after 7 days. And interesting, what they found is that the polymeric system, this cationic polymeric system, had a much higher expression compared to all the LNP, all the LNPs injected. Somewhat expected because it's a cationic polymer. It's and cell membranes in the body tend to be negatively charged, so these actually would be more prone to getting into the cells at the site of injection. And overall, yeah, we see almost 2 orders of magnitude difference in flux as far as luciferase, again indicating higher expression. There are some differences here between the lipids themselves, or lipids themselves, as well as a different helper lipid here, which we have lipid 3 with DSPC compared to DOPE. What we do see here is that these more destabilizing lipids, like DOPE, tend to result in a higher expression, which is expected compared to DSPC, which is used in the current vaccines, actually, in the current LNPs. So there is a little bit of a trend, but no significant differences. The only significant difference is here with this polymeric system. So the authors wanted to tease out really, you know, there is a huge difference in the expression levels. How does it functionally work? So they've encapsulated a hemagglutinin, flu hemagglutinin. Sorry, I see something in chat, but yeah, it's a flu influenza hemagglutinin strain mRNA, and they've done the same type of study where they would inject intramuscularly mice and then boost them after. They were boosted, and then the serum levels. So this is hemagglutinin protein expression was assessed after at week 6. At week 6 after injection. So every time 2 weeks after the injection, the levels were assessed. And what they found was essentially looking at IgG, so antibody neutralization here. So the functionality of these constructs when injected IM, what they found is that the antibody levels essentially were highest, actually, in the LNP groups compared to the polymeric group, which was interesting looking at the expression. These are expressed higher, but actually, as far as functionality, the LNPs worked better, especially this LM03 LNP here. And that really, it was a really interesting result for the authors, because it's also the fact that there were more data points here, but they're actually above the detection levels of the assay. So these antibody titers were just really, really high compared to what they expected and what they've seen before. This was a really exciting piece of data. So what the authors also moved on to do is to also look at really the protection on these mice after a challenge with the virus. So these were mice that were, after this regime of prime and boost, and then challenge with the virus, influenza virus in this case. And what they've shown is that all the LNP groups really resulted in a good protection of these mice as far as looking at their health by assessing their body weight. The polymeric group did result in a slight 10% dip in the body weight, indicating that, again, they were a little less healthy. For several days, they did recover eventually. And a naive group that did not receive any protection resulted in such a high drop in body weight that we had to sacrifice the animals 5 days after the challenge itself. So this is very encouraging, but this was, again, a model flu vaccines. What the authors are also interested in looking at a COVID antigen. So in this case, they would encapsulate an saRNA, cartridge encoding for a SARS-CoV-2 spike protein. And in the same type of study with a prime boost delivered intramuscularly, they looked at essentially SARS-2 specific IgG. So this is again functional antibody levels specific to SARS-CoV-2. And interestingly, they also compared to a group where the authors delivered 1 microgram of LNPs intranasally. So these all were intramuscular, like the current vaccines are delivered. The authors were curious if we could also see, antibody levels in the serum following an intranasal delivery. Here. So pretty high dose. Just to put it in the context of the Pfizer Moderna vaccines, the doses that Pfizer, for example, use in mice that correspond to the current human dose are around this range. In their studies, they've explored all the way to 5 micrograms with their modified but not self-amplifying RNA. And the dose that's kind of equivalent to the human dose is here in yellow or orange, I suppose. Here, the Moderna dose is a little bit higher. It's corresponding to around 0.4 micrograms in mice, I believe. So, and again, we see here that that's the dose that for the self-amplifying construct also results in a very good protection. And you see that protection actually plateaus for these, the 1 microgram group when injected intramuscularly. With this high dose, though, injected, delivered intranasally, you do see a protection that's, orders of magnitude lower, but comparable to maybe this group. What we also see from this data is that there's definitely potential in the ability to reduce the dose of delivered RNA, at least towards this group, which is a full order of magnitude less RNA needed. And again, if you're delivering less RNA, you're also delivering less excipients, which can result in a better safety profile. And again, less RNA just means you can make more doses in the same reaction volume with the same mass of RNA, if you may. So it's all beneficial, and we do see pretty good responses, all the way down to these levels, which again significantly higher protection compared to this polymeric system that at high dose performs similarly to something about 4 orders of magnitude lower as far as dose, looking just at SARS-CoV-2 specific IgG. So digging deeper into really functional humoral response to these antibodies, this is a SARS-CoV-2 neutralization assay, looking at utilizing the sera from these mice, looking at neutralizing a virus. So again, humoral response, showcasing again that these LNPs, even at lower doses, can provide quite significant protection and neutralization of a SARS-CoV-2 pseudotype of virus, really. Whereas the polymeric systems really don't do much in this case. Or at low doses, also we do see much lower neutralization of these viruses, and really the intranasal group also still seems to work after the boost, which is great to see. So following this, we've also looked at the cellular response, so like extracting essentially splenocytes, cells from the spleen, looking at T cell responses as far as protection. This is also a pretty standard assay, looking at interferon gamma production from these cells, and showcasing again that up down to 0.01 microgram of RNA in an LNP renders still a pretty good cellular response, T cell response to SARS-CoV-2 peptides. And I did forget to mention that, but this LNP is, just one of the lead LNPs, the best performing LNP that was chosen based on this previous assay. So this is the LNP3 with DOP used as a helper lipid. This best performing one here. That's the one that was chosen for all the subsequent SARS-CoV-2 experiments. Okay. So looking a bit deeper into really why there was this really vast difference between the expression level versus really functionality of these delivery systems, the polymeric delivery systems compared to the LNPs. The authors investigated, and this is really the last piece of data. They've looked into essentially a cytokine profile, an acute immune response, and what they've found is that across the different cytokines, as they were looking at them, there were no significant differences between these LNPs intramuscular compared to the polymeric system intramuscular compared to LNP intranasal, except for IL-6, and this is the only group. This is 4 hours after inoculation. This is the only really cytokine that showed any differences between this LNP intramuscular compared to the other experimental groups in this, and this might indicate that the LNPs themselves also provide some self-adjuvanting effect and help with the expression, not only expression. They help really to engage the immune system and to allow the RNA delivered and the spike proteins that were generated to result in a better protection which is something that we've also seen with the current Pfizer and Moderna vaccines, is that LNP systems on their own, they do have this sort of self-adjuvanting effect, so that the carrier itself can actually help potentially with the functionality of the delivered construct being used as a vaccine. It's quite interesting, but this is really the last piece of data I wanted to share with you. It's a quick update, and so we have a lot of time for questions, actually. Just to summarize, what the study has shown is that self-amplifying RNA is a viable modality for treating infectious diseases, showcased in this case on a model flu strain as well as SARS-CoV-2. And they are actually quite functional at significantly lower doses compared to the regular RNA vaccine, so potentially order of magnitude lower, while still being functional. And interestingly, expression levels of RNA, again, when we work with delivering RNA, delivering genes, delivering nucleic acids that would produce proteins, we always look at expression levels. And in this case, interestingly, they don't always correlate to the actual immunogenicity, so functionality of these vaccines. So it's important to also look at these other assays, the functional assays, to be able to assess how well your systems are performing. Expression, again, in this case, does not always imply correlate to the immunogenicity and protection. And then really showcased here is the fact that it's Precision NanoSystems in nanoassembler technology and lipids, allows for a end-to-end solution. These were made on the Precision NanoSystems instrument, and the lipids, ionizable lipids, were actually our proprietary ionizables that we've showcased here to be useful and functional in delivering an RNA vaccine. So just to give you some timeline really of how this collaboration with ICL happened and how it progressed. We've done a lot of work with ICL, and ICL already had some ongoing efforts as well as self-amplifying vaccine. So it's not something that was fully initially initiated, but it's a collaboration that played on both of our strengths. The GMP batch was produced earlier this year, and the clinical trials actually started in July of this year. So we're looking forward to seeing some of these results come out soon. So yeah, we were at half past the hour or so, I think, and we have time for questions.
Yeah, perfect. Thanks, Viet. Appreciate that that talk. Really interesting seeing a lot of that data, which even I've only seen in bits and pieces, I feel like, prior to this. So it's cool to see it all kind of come together into one data set. So thanks for doing that.
Great conversation, and thank you all for your questions and participation. So again, we couldn't do this without you, so thanks for your participation and support. And just a reminder, we hope you'll be able to join us on Friday, November 12th for our final Journal Club of the year. And I believe Viet will be presenting, and we look forward to seeing you at 10 a.m. Pacific time or noon Central time. So have a great day and the great rest of your week. Bye-bye.